Very good morning, everybody. It is my privilege to welcome you all to today's event on the behalf of Director of NAEB, as well as the Scientific Events Organizing Committee at NAEB. I extend my warm welcome to today's guest speaker, Professor Vidyanand sir. Today, we are organizing this event in the memory of Dr. Lalji Singh, dearly called as the father of Indian DNA fingerprinting. Some of us, the early recruits of NAB, were fortunate to have worked with him. We have listened to his advices basically during the SAC meetings. And I warmly recollect those days where, when there were different members questioning us, we were all very young and energetic, but we were just presenting a proposal. There were a lot of questions. And I recollect very warmly that he was having a calm demeanor, very silently, listening to us, very patiently listening to us till we completed our presentation and giving the most, expert advices which have shaped, I would say, the way that we started the labs. So I wove, some part of my career for him. So as early as his graduation days, he's very interested in science, and he started working on the cytogenetics of snakes. And he and his team had identified highly conserved DNA sequences in those days, as early as 1968, I'm talking about. And those sequences were later extended to other vertebrates as well as human sequences. And it was further used as probes for identification of specific source from where the DNA was isolated. Basically, it was used in forensic investigations, and many cases were resolved, which included civil and criminal cases, and some of which we are aware as well. So, for his contribution to Indian science and technology, in 2004, the government of India awarded him the Padma Shri Award. And he is also well known for founding many different institutions, such as the CDFD that we have here in Hyderabad in 1995, the Center for DNA Fingerprinting and Diagnostics, because DBT actually thought he would be the ideal person to, be the founding director of the institute with his expertise on fingerprinting. And as well as in 1998, 3 years immediately after CDFD was inaugurated, they had thought of looking at the assessment and also conserving the endangered species. And accordingly, the laboratory of conservation of endangered species, LACOONS, was started in CCMB in 1998. In 2004, after his retirement, he started a non-government, a non-profit research organization called as the Genome Foundation. And the sole idea he had his mind was to actually identify, diagnose, and also treat genetic disorders, specifically in human, from the underprivileged populations of rural India. Such were his high thoughts when it comes to the scientific, endure. And Dr. Lalji Singh had also served as the 25th Vice Chancellor of Banaras Hindu University, the same university from where he graduated with masters. And he also chaired the Board of Governors of Indian Institute of Technology, Varanasi, at BHU between 2011 and 2014. Before his term as Vice Chancellor, as I said before, he also served as the officer on special duty for CDFD, as well as director of CCMB between 1998 and 2009. He was truly a very great scientist whom we all miss today. And this memorial lecture is a day where we remember his life, celebrate his life, and also hear about his scientific contributions to the society. And with this, I invite our director to please come on stage and inaugurate the event now. Okay, sure. Let's pay our tribute. We'll have a 1 minute moment. Silence. Thank you, everybody. Ma'am, may I invite you to come on stage, please.
Thank you, Dr. Madhuri. A very good morning to today's speaker, respected Professor Vidya, esteemed colleagues, dear students, faculty, staff, and all NIV faculty. As we all know that Madhuri has already detailed out all about the great scientists whom we unfortunately lost in 2017. It was December 11th. It was very sudden. The whole scientific community was really shocked. So just in, since as she already told, he was our SAT chair. So within no time, this institute has decided, and it was very nice of governing body to permit to host this Lalji Singh Memorial lecture. And this is what minimum a professional institution can do for a person like Professor Lalji. And in 2020, the first lecture was started since it was a pandemic time. So this is the fifth lecture. The first lecture was arranged online. It was given by Professor Vijay Raghavan. That time he was PSA to Government of India. And after that, Dr. Shekhar Mande gave, then Dr. Partha gave, then last year it was given by Professor P. Balram. And this year we have Professor Vidya. Thank you very much for accepting the invitation and for being amongst us. I personally feel it's a very, very befitting lecture. It is going to be a very, very befitting lecture on this important, linked with this important occasion. Friends, as all of us know that the Institute celebrates the science done by the great visionary, I would call him, as everyone says, father of DNA, who always has shown his concern and was a bit displeased that India could not become a part of an international human genome project. A couple of times his own people have been saying. However, we decided that let us celebrate his birthday, which falls on 5th of July. So the lecture is usually arranged on 5th of July. But as this year it was, some commitments and a few other things also came. So we wanted a full attendance. It was I3C Brick PhD interviews. And this day has been chosen by the speaker, and we all have gathered here. So as we look forward for his lecture, and personally, I know him, his work, but I met him yesterday only. Had long chat on a lot of his ideas on scientific points, and his keenness to understand how things are in the Hyderabad Research Academic Cluster, NIAB, and so on and so forth. So I'll be very brief, especially for the benefit of this morning's guest who was honored us to be here. Just to throw some light, precisely this institute is 13 years old. It was May 2024 when we turned 13. officially entered to our teens. This was conceptualized and planned and given the shape by one of the former visionary leader at DBT, none other than the former Secretary Professor M.K. Bhan, who thought that animal biotechnology is a very important area and DBT should make a dent in this area through a very distinct way, not repeating what other animal science institutions are doing in the country. And that's how institute came. Professor Redanna was the officiating in charge, and this 100-acre lands where we are today. It was very gracious of the then Chief Minister and the then Vice Chancellor, Professor Syed Hasni. And that's how a little more land was given, but the institute is right now at 100 acres of area. After that, Professor Redanna, Dr. Subir Majumdar was the officiating director for full term, 5 years, before he superannuated and became distinguished professor. All the former directors in charges, they have really taken baton from each other and they have given their inputs not only to complete this concrete structure, but also at the same time they inculcated the academic and research vibes. For sure, no single person or a leader can really do all these things unless it is flanked by incredible people and people who are committed to the institution, that is a scientists, the whole administration, and students and fellow. We still have some of those senior students. I'm sure they're in the crowd who have really played a great role in making this institute. So another 2 minutes, I'll just make you know that what all we work on. So as our mandate and our vision is was discussed last evening during dinner. So the areas we work on, we focus on livestock, and some of us are now moving into those pathogens which are of importance, the equine origin, and some of us are going in a therapeutic mode using the canine origin work. But we work on infectious biology, we work on production biology. We have a good group of people in bioinformatics as well as in a nanobiology, wherein they work. And they work in sync with the scientists who are working in a core biology area to fulfill the mandated vision of this institute. The institute has a couple of important flagship programs. One or two we finished, and one which is going on is a One Health project. So I'm sure as you take a tour, some of our colleagues would be able to interact with you in all the wings. We do have a veterinary vaccine testing facility over here, which was put at place with using the Prime Minister Fund, PM Cares Fund. Which was made during the COVID times, and we are still kind of figuring it out to use it in a best use. So we have a good number of students touching 100 fellows and a lot of project staff with us, including the new PhD which BRIC has started, that is I3C BRIC PhD program. One student has joined us. So this is all what we are, and we would be interacting with you as we move on. Thank you so much. Thank you, everybody.
Thank you, ma'am, for introducing about activities of NIAB. So once again, good morning to one and all the NIABians who are present here with us. And it is with a great pleasure to have Dr. Vidyanand among us today. So Professor Vidyanand, scientific contributions are in the areas of developmental biology, theoretical biology, and evolution. They range over cell signaling, pattern formation, population genetics, and social behavior. He was also involved in demonstrating that cyclic AMP is amplified and relayed during communication in the social amoeba dictyostelium. He has shown that self-organization via cooperative signaling can explain the cell aggregation as well as the origin of multicellularity. That kinship is unlikely to be important for the maintenance of cooperation in this system. Together with Sharat Chandra, he suggested that imprinting may have co-evolved with sex determination and gamete dimorphism. He has used genetic algorithms to model phenotypic plasticity and evolutionary change. So Professor Vidyanand earned his BSc degree from University of Bombay and his PhD from University of Chicago. He held postdoc position at Hubert Laboratory, Utrecht, the Netherlands, Max Planck Institute, Tübingen, Germany, and Biozentrum, Basel, Switzerland. He worked as an assistant professor and later as a professor at the Center for Theoretical Studies, IISc Bangalore. He has been a member of the Molecular Biology Unit at TIFR, Department of Molecular Reproduction, Development and Genetics, and Center for Ecological Sciences, IISc Bangalore, and JNCSR Bangalore. So at present, Sir is an honorary professor at the Center for Human Genetics, Bangalore, and also as associated or affiliated as an associate at International Center for Theoretical Sciences, TIFR. With this, I would like to extend our warm welcome to Professor Vidyanand to join us. Thank you.
Dr. Sharma, members of NIAB, other visitors. I'm overwhelmed both by your kindness in inviting me to deliver this lecture, as well as by the hospitality with which you received me ever since I came here on a wet Sunday afternoon, which you did not let stand in the way of showing what NIB does, how it treats its visitors. Thank you very much. I'm especially conscious of the fact that this is supposed to be a lecture in memory of Lalji Singh, someone I knew, admired, and liked over the years. We were friends. We met on several occasions, both in CCMB and elsewhere. And I, in particular, was struck by the strength both of his personality and his originality as a scientist, his willingness to confront tough problems. The BKM satellite sequence that Dr. Madhuri alluded to was a case in point. In a sense, having caught hold of this problem, he never let go. And in addition to all these traits, he held on tenaciously to what he thought were the right sorts of things to do. And one saw this in particular when it came to his one saw this in particular when it came to his relentless efforts to build other institutions or foster the growth of other institutions which we believe were ancillary to CCMB and would go along with CCMB in furthering his dream of building molecular biology, modern molecular biology in India, which was on a par with the best anywhere else in the world. So Lalji will be deeply missed by everyone who knew him. And I think it's to the credit of NIAB that it's maintaining a memory of Lalji Singh by organizing these lectures annually. So once again, thank you for having me here. Now I begin with the theme of my talk, and I may as well tell you straight away that it's not going to be technical, it's not going to be detailed. In fact, if I succeed in making you think in a slightly different way than some of you may have been used to until now, my talk will have served its purpose. So right at the beginning, for the sake of those who want things in a nutshell, let me tell you what it is that I'm trying to get across. It goes as follows. Biology has a special place in the natural sciences. And it has a special place because it deals with living organisms, namely organisms that reproduce. Because they reproduce, they change in a very, very particular fashion. In other words, they evolve in a very, very particular fashion. And this is known as natural selection. The process of natural selection seems to be unique to biological organisms, which in turn lends an element of uniqueness to the field itself. This means that when we try to explain anything at all in the world, we have a certain way of looking at things and a certain way of explaining things, which differs between non-living entities and living entities. Because the principle of natural selection does not apply to tables and chairs and rocks and water and clouds, but applies to biological entities, to living matter. In spite of that, and this is true ever since the days of Darwin and Wallace, in spite of that, it has been apparent that there are several aspects of biological evolution, the evolution of living things, which have explanations which are not very different from those of conventional explanations in the physical sciences. So the question I want to leave you with at the end of my talk is it possible that there are ways of understanding the living world, especially the fact that it consists of entities that evolve? Is it possible to understand this in the same way that you understand the non-living? That's all that I want to try and get. In other words, are there places where natural selection perhaps takes a back seat to more conventional sorts of explanations that we are used to? So let me begin now. You see the outline there. The world is made up of things and phenomena. Living beings express certain properties which seems to put them on a separate plane. That separate plane is accounted for by natural selection. However, there are things which seem to take place without natural selection, and therefore they seem to put living beings also on the same plane as non-living beings. And finally, a very brief summing up. Right. In general, and this is a very, very broad truism, you can say that the universe, in fact, is made up of things, it's made up of phenomena. I categorize them under different heads, 1, 2, 3, and so forth. So on the one hand, you have things like clouds, water, rock. These are, of course, examples of our familiar solids, liquids, and gases. Then you have phenomena. Phenomena like rain falling, like lightning striking, like eclipses. Both these things and these phenomena are explained on the basis of what one calls physical law, some kind of law or the other. In other words, one believes that underlying them, there are deep principles obeyed by all entities The inverse square law of gravitation is one example. Newton's so-called laws of motion are another example. The laws of electricity and magnetism are another example. So one believes that there are such principles underlying the behavior of all entities, which account both for the things themselves, in other words, both for the fact that you have things like cloud, water, and rock, as well as for the behaviors that they exhibit, for the phenomena that they exhibit. Living beings, however, come on a slightly different plane. There too you have things, you have trees, you have plants, you have animals, you have, of course, microbes. Now, they are things of the same sort like clouds and water and rock. However, they seem to be in a completely different plane from them in the sense that they do things, and in particular, they do things whose explanation involves an element which one normally does not use in explaining the phenomena that are seen in non-living. So, for example, one's instinctive reaction to that first picture will be, there's a lioness trying to pounce on a zebra in order to eat it. There's a zebra trying to run away from the lioness to escape being killed. In other words, the explanation of the phenomenon that you see involves some kind of a purpose. You don't normally think of rain falling for a purpose, even though poets might think so and primitive human beings might have thought so. Today, one doesn't say rain falls, for example, to replenish the earth, right? Or lightning strikes to punish the wicked, or for Indra to show us his power. It happens because of natural law. Here, however, purpose seems to be a very straightforward explanation for what you are seeing. Similarly, the male peacock dancing has a purpose. What he's trying to do is to impress a female. So the explanatory framework that we use for things and phenomena in living creatures involves an element over and above what you use for explaining things and phenomena in non-living creatures. Of course, this peacock and that zebra and that lioness are made up of the same elements, the same materials as anything else. After all, if you look at the periodic table today, there are, I don't know what the latest number is, 100 and odd elements. So there's nothing you can get beyond that. It can't be that there's some special magic here which accounts them to do this, accounts for their behavior in this pattern. And yet, there's this notion of purpose lying behind. There's something else which seems to indicate elements of purpose behind it. And this something else consists of artifacts, namely things created by manipulating the environment in some fashion or the other. So that pot there that you see was probably made, we have to guess now, of course, either for drinking or for storing something. Those cave paintings at Bhimbetka, which are spectacular and move us and awe us when we see them there, were probably made, well, now here you have to guess even more, either by the painters to tell a story, you know, I killed this many animals yesterday in a hunt, sort of thing, or just for fun, right? Or simply to say, look, this is the kind of art that we can produce, which is what painters today might be doing, nonetheless made for a purpose. So you see, this notion of purpose is behind both the actions of living organisms as well as things which are made by artifacts. There's one special feature that artifacts exhibit, in addition to giving you the impression that they've been made for a purpose, which is that they're adapted to something. So that chimpanzee which is holding a stick in its hand, holding a twig in its hand, is not just holding any old twig that it found. This is a twig that it has cut off from a tree, that it has scraped clean with its mouth to remove any thorns that might be there, any leaves might be there, so that it's clean and smooth. Then it uses that twig to poke into termite mounds, annoy those termites, make them climb up on the twig, then pulls it out and licks off the termites. which basically using this twig as an eating device, which is exactly what that human being is doing with the spoon there in the next figure. So you see, artifacts of whatever kind they serve a purpose and they're adapted to serve that purpose, meaning there's something in the system that goes into building this artifact that over the course of time makes the artifact better and better suited for whatever the purpose is intended. In this case, it happens to be eating. So perhaps the very first spoon that was made was a very crude affair, maybe a bit like this stick. But then people realized that a hollow curve in the front would improve things, a long handle would improve things, etc., etc. So two features of living organisms are shared by artifacts, design and purpose, and secondly, adapt. Keep that in mind. By the way, there's a third kind of thing that one notices in the world if one looks around, which at first doesn't strike one as being the outcome of a physical law. However, it's a form of regularity which makes itself felt when you look at large numbers of the same kinds of things. So this is what one refers to as statistical regularity, which doesn't depend on any underlying physical law per se, other than the laws of chance, which are simply ordinary statistical principles based on probability. And if you go all the way back, probability is nothing else than the application of logic to phenomena. So the picture you see there is the distribution of porosities, porousness, in rocks. These are particular kinds of rocks, carbonaceous rocks. And this curve I picked up from some paper involving petroleum exploration, where people are interested in what kinds of rocks they're drilling through to get an idea of whether the yield of petrol is likely to be good enough. The point of the picture, however, is that the distribution of these porosities seems to lie on this smooth curve. You see that the actual measurements don't lie on the smooth curve, which is expected. After all, this is an experimental study. But statistically, those porosities which have occurred naturally, which have come about due to holes which have occurred naturally in this rock, as it has been shaped by the geology of the earth, those porosities are distributed in this very interesting, neat mathematical form. On the face of it, of course, that's a puzzle. Why should the rocks have got together one day and decided all of us will lie along this curve? But that's the way it happens. In fact, an even more dramatic example of the same thing happening is to do with the distribution of human heights. Obviously, with appropriate controls, sex match, age match, and so forth, human heights follow what's called a normal distribution. Again, a statistical regularity, which looks uncanny, which looks law-like, right? As if there's some deep principle involved behind it. Otherwise, why should this strange mathematical curve, by the way, also known as a bell curve or a Gaussian, all of us are familiar with this. Why on earth should these people have their heights falling according to these curves? Statistical regularity. So, we have seen that there are kinds of things in the living world, kinds of phenomena in the living world, which are explainable partly on the basis of their being material, like any other material. And partly it appears on the basis of something or the other which endows them with design and the capability to adapt, which clouds and water and rocks normally do not. So we've seen that. Secondly, we've seen that there are other aspects, other phenomena that you can see in the living world, which also seem extraordinarily regular, pattern-like, but which are originally based on statistical properties, not on physical properties. Evolution by natural selection, which comes to us from Darwin and Wallace over 150 years ago now, is the one principle in biology which managed to explain where these elements of purpose and design come from. Remember, the Darwinian theory of evolution came up at a time, certainly in the Western world, came up at a time when it was believed that life was on a different plane from non-living. In fact, you could make a very clean distinction between living and non-living. I remember even when I was in school, one of the popular questions in our middle school, I think it was distinguish between living and non-living. And it was a 10-mark question or something, because there were 4 or 5 points you had to remember. And if you just wrote down the headings and put the columns, you got 2 marks. So it was a good question in that sense. Darwin and Wallace showed that that distinction did not need to exist. That purely on the basis of materialistic arguments, without any invoking of religion, which was of course the strong underpinning explanation in the Western world, purely by means of materialistic arguments, using the properties of living matter, you could explain where these things came from, where these elements of design and purpose came from. Now, it's not my aim in this talk to try and give you that explanation, but that's today the matter of textbooks, and I'm sure all of you have come across it in your studies. That is simply a summary cartoon which indicates not really the Darwinian explanation per se, but what we now call the neo-Darwinian explanation, for the reason that there are elements to this explanation which Darwin could never have known of. So, for example, the existence of genes, the existence of particulate inheritance, the existence of mutations. It's a tribute to the genius of Darwin and Wallace that they came up with this explanation when they didn't know what there was inside living creatures. Even the cell theory was in its nascency in those days, let alone this clear-cut distinction between nucleus and cytoplasm, let alone this distinction between heritable factors contained within the nucleus and things elsewhere. Nothing was known. And yet they came up with such a brilliant insight into the workings of living matter that their writings are read even today. In fact, this is something interesting. Please think of it. People say, and I think correctly, that the one other figure in the history of science who can be justifiably equated with Darwin was Newton. You can come up with other names as well, but with Newton, there's no disputing. But Newton himself stopped being quoted probably about 100 years after his death or 150 years after his death. Not because he was disproven, not because it was shown he was wrong, but simply because Newton's ideas had been so thoroughly assimilated that Newtonian thinking was present in school books, in textbooks, as it is, of course, today, the way we learn about it. So people who write papers today in physics and chemistry, or at least in physics, won't write something and say reference Newton 1677 or whatever the date was. That's not true with Darwin and Wallace. Even today, you have papers on evolution written with citations to Darwin and Wallace. And the reason is there's so much richness in their thinking that every time you read them, you discover something new. So if I might give a piece of advice which may or may not be wanted, please buy for yourself a copy of The Origin of Species. The prose is superb. So read it. Don't read it at one go, but keep it by you. Read it in bits and pieces. And you'll see as it seeps in, the content seeps into you, you'll see how much richness it can. So anyway, so that's the principle. You have genotypes. With the help of the environment, you get phenotypes. Now something called selection acts. What is the selection? No mysterious force. It simply says that if individuals vary amongst themselves, variation is at the heart of the process of selection. If they vary amongst themselves, and if the variations are inherited, Darwin didn't know how, we know how. And if because of this inherited variation, some individuals are more successful than others at having children, then the properties in respect of which those individuals were more successful, the variations in respect of which those individuals were more successful will spread it. Extraordinarily simple principle. In fact, its very simplicity has had interesting fallouts, and two kinds of interesting fallouts. I just mentioned them. One, it began with Darwin's closest disciple, Huxley, T.H. Huxley. And that's a reaction which has persisted to this day among people who first study natural selection. Huxley is said to have said when he read Darwin's book, "How stupid of me not to have thought of this before him." The principle is that straightforward, that obvious when you think of it. In contrast to Huxley, there are many people all through the years, again including the present day, who say, look, the world is a very complicated place. This explanation is so simple, I don't believe that it can explain the world. How can it? There are many distinguished people who have taken that point of view. As we know now, of course, Huxley was right. It is a simple principle. Everyone should have thought of it. No one did until Wallace and Darwin came along, but we have it. Now, this is something which embellished the principle of natural selection, but I realize now it will take me probably too long to explain how it works. This was an embellishment by a person called Sewall Wright, who showed how natural selection could work in populations and move populations in what you might call phenotypic space. In other words, from a space in which you looked at their properties, move them from regions where their reproductive efficiency was low, or as we would say, from regions of low fitness, from regions where they move to high fitness. In other words, Wright set up a mathematical theory together, or I should say really in parallel with two other very distinguished evolutionary biologists, R.A. Fisher and J.B.S. Haldane, who at the time he died was an Indian. So we have some claim to pride in Haldane. Wright, Fisher and Haldane managed to put, you might say, mathematical flesh on Darwinian theory and showed how it could work in real populations. Wright's particular theory is known as the shifting balance theory, but I won't go into it in detail except to tell you, yeah, this is the way using genes, using gene-based phenotypes, in other words, genetic variations leading to phenotypic variations, using reproductive differentials, and using demography, population biology, could show how it works. And as I told you, the theories aim, as well as its success, was in explaining adaptation. The parts of animals and plants, here you see the feet of birds and the beaks of birds, the parts of animals and plants are exquisitely designed, or give the appearance of being exquisitely designed for a particular purpose. What is that purpose? Well, in one case it's acting as water filters and to feed whatever doesn't flow away with the water. In another case is to probe into holes and crevices. In some other case it's to, well, actually act as nutcrackers. In some other case to bore holes. In some other cases to tear flesh. So these are all sophistications of the spoon we looked at, except that these are spoons and knife and fork and hammer and chisel, but designed by natural selection, which is simply a physical phenomenon. It's not a supernatural phenomenon. So this was the achievement of Darwin and Wallace. So they showed how living organisms in their various attributes, including behavior, which I will not go into here, how living organisms could exhibit properties which made it appear that they had been designed by someone to fulfill a purpose. What purpose? Well, the purpose that you see, these kinds of purposes that you see. Okay, so far? I'll pause here because now I'm going to move on to rounding off my talk with giving alternatives. Are there any questions? Comments? So I assume there are none. We'll move on. Yep, there is one. Sorry. That's a fascinating question. Would you mind coming to it again at the end? In case I forget, please remind me. That is a fascinating question. Yes, please. No, this is the whole point. So the idea there is, let's go back to that heading first. Right. So the idea there is no long range planning in natural selection. Right. So there's nothing in the process of natural selection. If you go back to one of those bird beaks, let's say woodpecker's beak, which actually works like a hammer, a pointed hammer. There's nothing in the process of natural selection which, now if you go back in time to the long, far back ancestors of today's woodpecker, there's nothing in the process of natural selection which would tell them one day this will have to end up with your descendants being very efficient hammerers into trees. We cannot. We cannot. It has happened. This is the whole point. We cannot predict that as far as we know. Come again. No. This is the whole point. So the whole point of natural selection is that without any long range planning, without any long range aim, something results shaped purely by contingency, shaped purely by what works now. So it's very much a hit and miss affair in the short term. Exactly. 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 Precisely. That is the beauty of natural selection. For those of you who are interested in metaphorical language, natural selection is like a random walk. Have all of you heard of a random walk? But not simply a random walk, because a random walk takes you nowhere. A random walk with a bias. Without a foreordained direction. Without a foreordained, the important thing to keep in mind is in hindsight, of course it's direction. The human eye is a beautiful example, starting from presumably some kind of semi-transparent pigment epithelium to come to this incredibly complex device for focusing light. It looks perfectly directional. Yet step by step, when you try to follow it, you see that what has been happening is that at every step, things have improved in terms of improving the reproductive efficiency of the carrier by making these small changes, by ensuring the success of small change. So natural selection always works only in the short term. It's very much, one word which is used often by students of evolution is opportunism. Natural selection is a supremely opportunistic device. Anything that works now is fine by it. Yeah. Exactly. Exactly. Adaptation is the end consequence of natural selection. Yeah, please. Yes. One moment. CRISPR as carried out in the lab or CRISPR as carried out by the inside the cell? By the okay. No. So what that shows is how experience can play a role, right, in furthering natural selection. So I'll give you a simple example. We won't go to CRISPR, something very, very simple, which was discovered by Chow and Cox some 30 years ago. I'll take 5 minutes if you don't mind, just over this. Okay. You have a bacterium sensitive to an antibiotic, right? It can't grow in the presence of the antibiotic. There's a plasmid which carries an antibiotic resistant factor, okay, gene. If you introduce this plasmid into the bacterium can grow in the antibiotic, but it's become resistant. Standard things that you use whenever you do transformation and selection in the lab. Very good. What Chow and Cox did was to grow this plasmid bacterial association in the presence of the antibiotic for a great many generations. Many thousands of generations. Luckily, you can do that with bacteria. At the end of this many generations, they cured the bacterium once again with the plasmid. And now ask the following questions. How does the bacterium by itself and how do the bacterium plus plasmid combination do both in the absence of the antibiotic and in the presence of the antibiotic? In the presence of the antibiotic, the bacterium still needed the plasmid, of course, because the resistance factor still was on the plasmid. The curious thing was that in the absence of the antibiotic, the bacterium plasmid combination did better than the bacterium alone. You see the point that I'm trying to get at? So during the course of, this was not natural selection in the way we use it, selection carried out in the lab. But as far as the bacterium is concerned, it doesn't know who is doing the selection. It doesn't distinguish between Cox and nature. So what is happening over this course of forced coevolution between these two reproductive elements? Now we'll think of them as two living creatures, just for our sake, which are part of one big living creature, the plasmid and the bacteria. During this course, secondary changes have taken place opportunistically. Mutation is a purely chance affair. Secondary changes have taken place opportunistically, which have strengthened, if you wish, the bonding between plasmid and bacterium. They have nothing to do with the antibiotic. It so happened the antibiotic was there. So things like this can happen, which give you a Lamarckian air. But it's not Lamarckian, it's perfectly. I'll show you an experiment later which looks even more Lamarckian, but this is not. I shouldn't overshoot my time. Okay, so we've been through that, adaptation, right. Now, yeah. Yeah.
In that case, when we talk about recover the purpose, basically when you have a mutation.
Yes.
So you're looking to recombination.
Yes.
Those are all randomly.
Yes.
But now figured out the relational terminology becomes hotspot.
Yes.
So what is the purpose of hotspot?
Yes. Now, one brief thing. I won't go into this in detail. Again, it's a very good question. The word random is used in a particular sense in biology, right? We talked about random walks before. This random is not the same as that random, but both are random in a certain sense, meaning that they are chance. In biology, random simply means this. So a random mutation simply means this, that the consequence of the mutation is independent, right? Has nothing to do with the likelihood of its effect, which means a favorable mutation can occur with as much probability, as high a probability, or as low a probability as an unfavorable. So random with respect to direction is what. There is nothing which predisposes organisms to mutate in a favorable direction. Get the point? By the way, those of you who have studied bacterial chemotaxis can sort of sense a formal similarity between these two, but there's a difference there because the bacterium changes, there's not mutation of course, but these are what are called tumbles, changes in direction, changes its tumbling frequency in order to maximize its chance of reaching the goal, which is food. That is not happening here. So in that sense, that okay, rapidly. Good. So what is it that I'm going to tell you now? And here, if I run out of time, there's no problem, because I'm just taking you through a series of case studies in which evolution seems to take place, is apparently taken place. Obviously, we mean here biological evolution in living creatures, but without natural selection. And all of that I'm going to illustrate very rapidly in these sketches is how it could take place. In other words, what the underlying explanation could be. The simplest, of course, is statistics. You don't even need the physical law for that. And this depends on the discovery that we have learned to live with today, that mutations come in all sorts of colors and shapes and sizes and so forth. What do I mean by that? I mean that mutations can be categorized in several ways. One of those categorizations is according to whether they are advantageous or disadvantageous or neither. And this was something no one had foreseen until about 50 years ago. Today, of course, we live with the knowledge that a great many mutational changes in the genome are simply neutral, neither advantageous nor disadvantageous. And as Kimura showed when he made this proposal 50 years ago, and as a great number of experimenters, among them Linus Pauling, for instance, Zuckerkandl, many others, showed the consequences that some places in the genome mutate really randomly, meaning that they're neither advantageous nor disadvantageous, has very profound implications for evolution. And in fact, Kimura pointed out very early that because of this neutrality phenomenon, certain genes, meaning genetic variations, that did not have any effect on the phenotype. Why? They were neutral. However, these variations could spread in a population. Normally, spread in a population means because of natural selection, because of reproductive advantage. Kimura said you don't need either of these two, and they can still spread in the population. How? By sheer chance, statistically. And here is some amount of mathematics which I won't go into here, which tells you what the probability of spreading to fixation is. Turns out to depend on whether the mutation is strictly neutral, slightly neutral, very slightly advantageous, and on the population size. Yes, please. Yeah, certainly. Quite right. Okay. Now, one very interesting consequence of this, extremely interesting consequence of this, which Kimura brought out. You can have a mutation which is advantageous. Therefore, natural selection predicts it will spread in the population. But it can be lost if the effect of chance overwhelms that of natural selection. Therefore, for an advantageous mutation eventually to spread and evolution by natural selection to take place, it has to take place many times. Many trials, one may succeed. So this is a completely different way of looking at natural selection. It's not a deterministic process in the sense we had thought of, but requires first the establishment of a foothold by the mutation, and then, of course, equally surprisingly, you can get disadvantageous mutations, harmful mutations, which can spread again by chance. So these are two things which completely overturned, I can certainly say, all our thinking in biology, turned things upside down. Okay. It had one other consequence which was very, very strange, and it's worth explaining, but unfortunately, shortage of time makes it impossible. So I'll simply state what it was. This consequence is that if you look at neutral changes alone, and therefore evolution by neutral changes alone, and therefore try and measure evolutionary rates by seeing how many changes are accumulating as a function of time. I hope it's clear what I'm trying to get at. You can do that by building phylogenetic tree. People do this all the time, of course. Then the prediction of Kimura's neutral theory is that these changes should accumulate linearly. The rate of evolution should look like a straight line, but not the same straight line, because the straight line should have a very steep slope in the case of proteins or DNA sequences, which can evolve very rapidly, and a very small slope in the cases of those DNA sequences and those proteins which have such rigid requirements on that functioning that they cannot evolve rapidly. Very easy to think of these differences. So for example, if you have a blood clotting factor, well, if it doesn't work, you are in trouble if you bleed and so on, but otherwise you can lead a normal life. Right? It's not a protein which is vital for your health under normal circumstances. On the other hand, if you think of a histone, almost certainly any mutation in it dooms you. In fact, there is no you. It probably is an early zygotic lethal. Right? So these two proteins, both of which evolve at a linear rate according to the theory, if there are neutral elements in their DNA sequences, evolve at very different neutral rates. And this particular idea of a linear rate of evolution of proteins came to be called the molecular clock, and it's reasonably well established now. Now, as always happens, the earlier findings showed very nice linearity. Now, when you probe things more and more carefully, the linearity is not so nice, but nonetheless, the basic point remains that different proteins evolve at different rates. And those rates seem to reflect the degree of importance that those proteins have for the well-being of the organ, for the life of the organ. So, brilliant example of evolution at the molecular level, not at the organismal level, remember, this thing of protein, which works in a way in which natural selection seems to take a backseat. Detailed consequence of that theory, which I'm going to skip over here, again in the interest of time. Now, many other sorts of ideas which have come up, which depend on something like neutrality, not the original neutral idea of Kimura. So, for example, it's well known that gene duplication is one phenomenon which has acted as a kind of fuel for evolutionary change. By now, this is well established. Okay, so what does gene duplication give rise to? In the first instance, obviously, a second copy of a gene that you have already, which is therefore redundant, right? Why is it not thrown out? Well, if it exacts too much of an energy load for DNA replication, maybe it is thrown out. But if it doesn't, it will hang around. So it can stay as a quasi-neutral element. What can stay? The duplicated segment. Can stay as a quasi-neutral element, hanging around, so to speak. Now, building on that, all kinds of things can happen later. Of course, the simplest thing that can happen is that the duplicated gene becomes a pseudogene. So it's gone. It doesn't do anything in terms of protein synthesis, certainly. But there are all kinds of other things that can happen, which this slide and the next slide illustrate. Again, I won't go into details. You can read what the slides say. So, for example, you can get mutational changes in the duplicated copy in an essential gene, which normally, of course, would have led to lethality. But because you have a spare tire, so to speak, in your car, this puncture doesn't matter. Right? Having undergone that genetic change, which normally would be lethal, you can now, meaning the organism, can undergo a second genetic change, which complements this first one in a way that it makes them able to function in some new fashion. So you say this is very interesting. It gives you a stepping stone to something you could not have achieved without that duplication. Right? But that initial step, which was the duplication itself, was a chance event, nothing to do with natural. The word opportunism has cropped up here again. Now, many of you who carried out experiments in the lab have seen this. In a great many cases, you mutate a gene, turns out to have no visible effect. You use the word null phenotype. Right? On the other hand, you mutate some other gene which codes for some other protein, it turns out to have an effect on the trait that you are looking for. Why is that? That's because many proteins seem to function as heteromers, where the second copy, so to speak, meaning the partner copy, complements the function of the first copy. How does this complementation come about? Again, a possible way, not involving natural selection for the first step, and this is simply because proteins have only so many, let's say, binding affinities. So if you have a very thick soup of proteins, which may well be the case inside the cytoplasm, protein associations may be occurring at random all the time, simply by mass action. And if such associations occur, it's very likely that one particular association, if it recurs time after time, can lead to secondary genetic changes, like that plasmid bacterium experiment that I gave you, can lead to secondary genetic changes, which can now fix the change when it's advantage. So these are imaginative thinking suppose, but what they help is to enable you to see how something might happen, which on the face of which is a bit hard to understand. Again, I completely skip any attempt to explain this slide, but I find it fascinating because this was brought up by a person called Bahram Hushmanzadeh. This is an example to show how neutral evolution can also lead to social behavior, cooperative behavior. Fantastic kind of example. Whereas the conventional theory would say that cooperative behavior can evolve only when you have relationships, when you have close relatives. This shows how you can get away without that. A person called John Bonner has gone to the extent of saying that entire organismal phenotypes may be neutral. You should read his book to see this. It's really a booklet, not a book. So it's extremely readable. And he uses marine microorganisms, diatoms, foraminifera, and so on to illustrate his point. And his basic thesis is that these tiny organisms are so many, and their life cycle is so simple, right, that they are tolerant of many more genetic changes than higher organisms like us are. So, for example, it's taken for granted that with human beings and animals, most early acting mutations are liable to be harmful, right? And this is almost a rule of thumb one uses. Whereas here, most early acting mutations, according to John Bonner, are probably neutral. So there's a lot of phenotypic space, you might say, available for them to explore, right, without running into other risks. Whereas our genomes are so complicated that whereas it might be advantageous from the point of view of vision to develop a third eye, the number of interactions involved within our body as it is would make it almost guaranteed to be a lethal change. I have the spirit of what I'm trying to say, etc. Okay. Other cases, this is the most fascinating one for me, and come to think of it, I think I'll end with this because time is running out. How much do I have, by the way? No. This is a thing which people always tell. 10 minutes. Okay, so I assume I have 5 more minutes. This is my favorite example. Example of what? Example of how evolution might have taken place in something very fundamental without natural selection, involving just ordinary law, physics and chemistry is what I think I've said there, or said in the previous slide. This has to do with the genetic code. All of you know the genetic code. All of you know there are 64 codons, or 64 minus 3 codons, which code for an amino acid. And all of you know that the way the code works, right, is by means of this remarkable creature called tRNA, which at one end recognizes a triplet codon, and at another end recognizes an enzyme, a synthetase, an amino acid synthetase, which brings a charged amino acid and sticks it on. These two ends are this form, that form, a molecular standard, almost 50 angstroms. They can't talk to each other. Figuratively, of course. 50 angstroms is a huge distance by molecule. 50, 60 angstroms, I don't know. So the feeling has grown that the genetic code is purely a formal device for, as we say quite often, to go from the language of DNA to the language of protein, the language of nucleotide triplets to the language of amino acids. Purely formal. In fact, there's a famous phrase due to Crick, in which he said, the genetic code is a frozen accident. It's there because it works. Once it worked, changing it was too serious a matter to contemplate. Very much like human language, in fact. All our languages are in this sense, with some history thrown in, frozen accident. The fact that I'm speaking in English here rather than Telugu has nothing to do with the relative fitness advantages of English versus Telugu. Entirely accidental. But turns out, so this is the tRNA in a cartoon form, the familiar cloverleaf thing, the anticodon at the bottom, and the acceptor stem on the top, where the enzyme lies. Turns out, and this is fascinating work done by Schimmel and colleagues, turns out that if you take the acceptor stem alone in various modified forms, you can see 3 modified forms here, and pay attention to some details. So, for example, in their paper, they say, by the way, you see that it mimics in some way a tRNA molecule. Okay. Now, you pay attention to, oops, wrong. How do I get spot? It doesn't matter. I can tell you from here. If you can mimic some things accurately, for example, you can see the N73. Those numbers, by the way, correspond to the usual numbering on tRNA. This is much smaller than the tRNA molecule, of course. So if you keep that N73 conserved, and you're careful about 3 or 4 bases below that N73, then you can get pretty accurate, not foolproof, charging by a cognate amino acid synthetase just by this active stem alone. There is no mRNA, there is no anticodon. But this fragment, nucleotide fragment, if you wish, is capable by itself of recognizing what within inverted commas you would call the correct amino acid. Now, you have to take this with a few caveats. The most important caveat being the recognition is not foolproof. It makes mistakes all the time. It's a sloppy process, but nonetheless it's selective. It doesn't say, okay, any amino acid is okay with me. Selective for a particular amino acid. And that depends, as I said, on those 3 or 4 bases which are below the infinity. So purely physics and chemistry, so this is now our thinking, Schimmel's thinking. Purely physics and chemistry may have made it possible for the interaction between whatever you call this creature and an amino acid. So you could have amino acids strung together, you still don't have a peptide bond being formed and so on, so we'll wait for that. Okay, but nonetheless, you could have the selective picking up of amino acids entirely without a genetic code. This is a wonderful kind of thing. By the way, this story has gone on much further. I'm using this old slide because it makes the point. Theories on a physico-chemical basis for the evolution of the genetic code have gone on much further. But now they seem to be, pretty much becoming accepted. But the genetic code was a later add-on to what was there already. So I'll finish my talk here. I'll just sum up things in a few words. I haven't answered your question, but I'll come to that since you'll take it up. Too much. Yep, summing up. Right. So the picture which is building up now is that the kind of explanation that you have for these things on the right, which are very varied, have drastically different properties, are creatures, one might say so, with very different phenotypes, but are nothing else than carbon atoms strung together in different ways. One of them is diamond, one of them is graphite, one of them is soot, one of them is C60, one of them I think is C120. There are nanotubes somewhere, I think, or there should have been nanotubes. Okay, all kinds of things. All of these are just carbon atoms, but exhibit these dramatically different properties. So that if you were to look at them purely morphologically or purely phenomenologically in terms of the properties that they exhibit, you say these are different things. But they are simply carbon atoms thrown into a test tube, shaken about, and they give you this. I'm being loose, of course, in my language. So the metallurgy behind preparing them can be tough, except in the case of C60, which amazingly enough was being made in homes, God knows, ever since human beings invented fire, because it's part of soot. It was right there. No one knew it was there. You know, smoke, what do you call it? Coal fire soot. Anyway, so the claim now is this kind of shaking up in a test tube business and waiting for things to happen might also be partly responsible for the things on the left, which all along we've been led to believe need this extremely special explanation called natural. Obviously, no one is throwing natural selection out of the window. It's impossible to do so. Adaptation hits you in the face if you try to think of it. So someone has to come up with a good physics, chemistry based explanation for adaptation. No one has so far. So natural selection does remain the default option for understanding life and living organisms. And yet it appears that it can be pushed in its way by physical force, ordinary physics, ordinary chemistry. And deepest of all, I think, is this difference in explanatory paradigms. The explanatory paradigm that we use normally in our day-to-day life, by which I mean life in the lab or in the field, whatever research all of you are doing, the explanatory paradigm that all of us use is an ahistorical paradigm. Ahistorical, which means what? Which means that if I take this and drop it, it falls down. It simply has to do with the fact that I dropped it. I could drop it somewhere else, some other time. This could have a different shape. Doesn't matter. It's the falling that counts. To give you an amusing example, like a friend of mine once thought up, an apple falls on someone's head. His name happens to be Newton. Why did the apple fall on his head? The ahistorical explanation, which all of us believe is the correct explanation, is that as it ripened, it grew heavy, the twig grew lighter because sap started stopping supplying it with nutrients. And when the weight got too heavy, it snapped. That's it. Nothing else. Same kind of explanation. In fact, exactly the same kind of explanation for why the moon goes around the earth. Same kind of explanation. No special reason. But you could think of a second explanation, a historical explanation, which appears laughable, but on the face of it, why not? And the historical explanation would be, in the old days, long, long ago, in Puranic times, let us say, in our India, apples flew all over the place. They didn't fall down. Some fell up, some fell to the side, some fell here, some fell there. But only those that fell on the ground took root. It's only there that the seeds found the nutritive soil environment where they could grow. So natural selection over the millennia left us only with apples which fell down. nonsensical, of course, but not within its own terms, logically impossible. But that is the kind of explanation which comes to us naturally, thanks to Darwin and Wallace. Well, that slide has disappeared for the slide which you saw on the left for living organisms. That's a historical explanation. So the essence of the point that I've been trying to get across today to you is, to what extent do ahistorical factors play a significant role in the evolution of living matter, not non-living matter in this case. Thank you very much for your patience, and I am sorry for taking up your time. Before anyone else, can I take up your question? In fact, I have a vested interest because I had a slide for it. Two slides, in fact. So this is a case, please tell me if I'm answering your question, I'm not sure, where you seem to get a phenotypic change before a genotypic change. And this is a phenomenon called genetic assimilation, and it's tied up with a second phenomenon called canalization, which was much in vogue among, in fact, plant and animal breeders a long time ago. How many of you in the audience have come across these terms, just for curiosity? Canalization and genetic assimilation? You have. Okay, a few people have. Anyway, the idea is very simple. So the experiment illustrated on the screen was carried out by a person called Waddington, where he threw fly pupae either into a jar containing ether vapor or raised their temperatures very high. Basically gave them what you would now call an environmental shock or an environmental insult. Very strong shock, very strong insult. Most of the pupae died. The few that eclose give rise to adults which look, well, let's say in bad shape, pretty bad shape. All sorts of deformations. And among those few, there are a few whose body seems to phenocopy the phenotypes of known genetic mutations. So Waddington in particular, in this experiment which is illustrated, was looking for flies which eclose to give rise to adults, in which the adults seem to have, in addition to the normal two wings, their usual halteres, which you remember are the structures behind the wing, also growing more than normal. They have not become wings, but they look like overgrown halteres. So the halteres are that can't even be seen. Now what Waddington, please remember, only environmental shock. Now what Waddington did was to breed, carry out sib mating between the progeny of those pupae which gave this particular outcome to an extreme degree. Carried this out generation after generation. Remember, this is an experiment which involves selection, but as far as you can make out, no genetic change. He's not applying a mutagenic agent. Heat shock is not a mutagenic agent. And to his amazement, after almost a handful of generations, I think the numbers were 21, 22, 24, something like that, he started getting flies which bred true for 4 wings. And in fact, the fly on the right, believe it or not, as you see, it looks in pretty bad shape. The fly on the right is one of those flies. You see only 2 wings because the normal 2 wings have been taken off by Waddington to illustrate this. So those are the 2 halteres which have grown to wing size. In fact, they mimic phenocopy, as I said, the fly on the left, which is a genuine genetic mutation. It's called ultrabithorax, for those of you who know, 4-winged fly. So the point here being the following: Waddington seemed to carry out an experiment whose outcome was Lamarckian. There was an acquired trait this tendency of the Haltiers to wingness, and that acquired trait got reinforced over the generations. Of course, it turns out that the correct explanation has nothing to do with Lamar. It was given by Waddington himself, but it has to do with something very interesting, which touches on another theme that we picked up. Namely, when you are trying to select for flies with larger and larger Haltiers, which Waddington was doing, you are carrying out two kinds of selection. One, visible, what you see. You are picking from flies which are larger and larger Haltiers. So, if there was any genetic variation, for genes which were in the Haltier morphogenesis pathway, you might have been selecting for them. But unknown to you, you are also selecting for regulatory genes, which make it easier and easier for this switch from Haltier to wing to occur. In other words, you are lowering down the threshold for this transformation in terms of the environmental insults that you're giving, ether or temperature. And if you lower down far enough, if the threshold goes to 0, you don't need it anymore. Huge change in the fitness. These are sick flies. It's just a morphological thing. Huge change in fitness. Beg your pardon?
That's a clear indication of.
I'm sorry. Why? No, I didn't get that point. How do you mean?
Since the fitness is the though there are no genetic alterations, but still the fitness is changing.
Yeah, I didn't get the human life link.
No, when I talk back to environmental effects
Yes.
of the human.
Yes, yes.
So, though we may get adapted and further continuously,
Okay. So in the human context, you could well imagine the following, which is any drop in fitness could be compensated by improved technology. Please keep that in mind. Technology is one way of keeping natural selection suppressed. All along. By the way, I wear spectacles as well, like you. We would have been in bad shape many thousand years ago. You get the point, right? So this fly is no good in the lab. You have to sort of almost force feed it. Keep it going. Nice of you. Well, that needs another talk, but I'll say just two things very briefly now that you've raised them. One, if you're thinking of RNA viruses, they are very different kinds of beasts from everything else you know. Primarily because they have RNA genomes, and RNA genomes turn out to be extremely poor at error correction. So there are two things really. One is extremely poor at error correction, and secondly, extremely tolerant to change. All of you know this. I suspect many of you probably are working with RNA virus. Okay, so these two features mean, as it is even with us, even with this E. coli which you grow in a test tube and think you're cloning, even their genotypes are not identical. You know this, they vary in a matter of time. But with RNA viruses, genotypes vary so much that even for what you would normally call the same virus, it's the same because it probably causes the same infection. If you take an EM picture, it looks the same. This is one, it has the same host specificity, all of those things. But if you look at the same virus, genetically, the genome seems to be very, very, very different. In fact, people now call about genomic, use the phrase genomic cloud. So it's not a genome, it's a cloud. Okay, so that, I don't know whether I answered that part of your question. Second part. So the evolution of RNA viruses is an extremely interesting subject because it involves an organism of a different kind. Secondly, why do viruses evolve in a way that makes them less infectious? This is a fascinating question. I'm tempted to say you should not have asked it because it relates to the area of my study, which is social behavior. The point is the following. Here you have a virus, spreads from host to host. And now we'll try and use slightly anthropomorphic language. Please forgive me. The virus wants to replicate, spread itself as rapidly as possible. The host, of course, wants to stay healthy. So there's an evolutionary arms race going on. On the one hand, in the virus, trying to make it more and more infectious. On the other hand, in the host, trying to make itself more and more resistant. Usually, in the short term, viruses win these races because they evolve much more rapidly than we can. Our life's generation time is what, 20 years, 5 years. Whereas for a virus, it could easily be hours. Okay. Now, here's the catch, however. Suppose the virus becomes so infective, right, that it starts killing the host very early in the infection. What's going to happen? The viral titer that has developed in the host is still small because the host has died. I'm assuming that once the host is dead, the virus doesn't have any source of nutrition. So this is where the selectionist argument, evolutionary argument comes in. Think of two viruses. One virus which is like a temperate virus, like a temperate phage, hangs around with the host. Maybe lyses every now and then, but goes back again, gets integrated, hangs around. Over a long period of time, it will accumulate a high titer, right? The host will also survive. Therefore, the defense mechanism of the host will not be as active against the virus. That virus, which is in this sense behaving in a conservative way, not going out for all-out attack, is more effective at spreading its genome than this highly infective, lethality-causing virus. So over the course of time, by the way, the argument is very general, applies to infectious diseases in general. If you don't do anything to them, chances are that they will gradually go down in terms of infectivity. That's the crux. By the way, for those of you who are interested, since you're working on all of you on animals, please look up. There's a fascinating account of this with the myxoma virus and rabbits in Australia. Please read it up. There's a huge amount of literature documenting this. Yes, please. Yeah. I'll go back. Yeah, please tell me. Yeah. Okay. Let me just make this clear. And I think the person who asked this question previously from here made the same point. The word purple is being used here in the same way that we would use the word in ordinary language. Right? So let me give you an example. Let's look at this auditorium. Why is it here? Well, very simple answer. Lots of carpenters came, put together wood. The people came and brought these chairs and put them there, and someone fitted lights and this and that. And that's the explanation for this auditorium. The second explanation, which is on a completely different plane, is that the auditorium is here because someone wanted a place to have meetings. That is the purpose. So the idea behind having an auditorium, say, existed much before the auditorium itself. That is purpose. That does not seem to happen in non-living matter for sure. Sure. But seems to happen very much in the case of living matter. All the time. All the time. You go to the cafeteria for the purpose of eating lunch. But the reason for your action pre-exists the lunch itself. It's not stimulated by lunch. It's something internal to you. I mean, you can think of 101. I showed 2 slides there. The lioness attacking the giraffe and this and that. So the point here being, if you take it as an assumption, which all of us do, I think, if you take it as an assumption that life is a material phenomenon when you get down to it, therefore has to be ultimately explained in the same way you explain other things in the world. Can you somehow rig the laws of nature or the rules of nature so that it seems to bring in this element of purpose? This is what Darwin abolished. They said you don't need to think up anything supernatural, no black magic, but there are ways in which natural laws can work whose outcome seems to be that there's purpose in the living. So it's in that sense that it is the world. So the environment, of course, has challenges, right? And those that can overcome the challenge survive. Those that don't. But a matter of overcoming or not overcoming, thing of not overcoming or overcoming is entirely determined by chance. Well, I mean, if you use the word, but that's not the usual word, usual sense in which one uses the word. See, okay, let me put it this way. The aim of the explanation is to try and account for living creatures as material objects. If you say the bacterium already knows that it has a purpose, which is to give rise to other bacteria, that's one thing. On the other hand, if you say the bacterium is a bag of chemicals of various kinds, and that bag put in a certain environment goes through various physical chemical reactions which give rise to 2 bags. There are 2 different points of view. The second point of view has nothing to do with purpose. The first point of view says, yes, the bacterium says I've been put here on earth to make more bacteria. Purpose. But that is not an attitude we take. So this overcoming the environment is something that happens. Something that happens. But on account of events which do not have a design, which are not planned. Which is why I showed the example of this artifact. So assume that long ago, before there were spoons, I just showed that as an example, people took pieces of metal and beat them in all sorts of random ways. And then one of them turned out to be spoon shaped. In the case of artifact, you wouldn't accept it, but that seems to be what has happened in the living world. There was one question here, right? Yeah. Thank you. Now I need to think of your question a bit. So you're thinking of peptides, for example, which contain the amino acids. That's right. Peptides and things of that kind. Right. Now, as far as I know, all of these are based on enzymatic synthesis, not on the basis of having a nucleotide triplet that's specified. Am I right? Right? So they're just, you can think of them as any other molecule, any metabolic product. So they are not on any different plane. They are amino acids, of course, and they are d-amino acids, so their chirality is unusual. But as far as we are concerned, we can view them just as any other small molecule. You have ATP made through a complex biochemical pathway. So isn't it? So yes, please. No, not only a good question, but an important question, because based on where we are headed, it's clear that humans have affected natural selection already to a considerable extent. Many species have gone extinct because of us, thanks to us, you might say. And it's also equally clear that we have staved off the effects of natural selection on ourselves. That's what all doctors are doing. They're trying to fight natural selection. And that's what I was trying to say earlier. Technology is a big enemy of natural selection. Now, what it will do in the long-term future, I don't know. I find it fascinating. There's a person called, what's his name? This person who's written these books, Homo Deus and Sapiens. Harari, thank you. Harari has very interesting speculation. So he's one of these people who thinks that artificial intelligence is not just going to take over. In some respects, it's already taken over, which would be nice, in the sense it now tells you a completely different form of life. I like to call it life, but that's what it is for all practical purposes, which may now start evolving by natural selection, not because of what you do. But you should read Harari for his speculations, very interesting. Let me just interrupt. Your first point is perfectly well taken. Go along with that. Okay, I can only give you a very personal response to that. I can't claim that this is sort of, a general consensus in the field or any such thing. personally, I find it extremely difficult to come to grips with the notion of consciousness. So like all of you, I imagine I've thought about it, I've read about it as much as I can. I have not come across any, I won't say definition, but any set of thoughts which enable me to put my fingers around. I can't hold them in my hand. So I find that very difficult. For my purpose, again, personally, I would say that what we think of as consciousness is a reflection of internal states, right? Which are a reflection of the physico-chemical goings on inside these states, inside these entities, whatever, signaling between neurons, short-term synaptic plasticity, reinforcement of connections and so on. Now, to some ensemble of these things, we give the name consciousness. What that ensemble is, I don't pretend to know. I certainly don't understand. So that's one of mine. Secondly, I can give a better answer to the second part of your question. I don't think any change in any entity that you think of as consciousness would make it think this far ahead. By the way, human beings are an exception in the following sense. All of you who work with animals know the following thing. In general, in biology, the genotype of an organism determines its phenotype pretty much directly, obviously with environmental influences, past history and so on. Normally, the genotype of an organism does not influence the genotype of the offspring of that organism or grandchildren, so on, normally. However, we also know since a great many years that there are things called maternal mutants, maternal effects, paternal effects. So that happens as well. We know this. So we have a genuinely genetic basis, understandable basis for the transference, right, of phenotypic information, which is the type of the genotype, across generations, jumping generations. So the point that I made earlier about evolution thinking only in the short term has to be modified to that extent. Now, this is the interesting question. Can you think of possible influences like this jumping not across one generation, but 100 generations, 1,000 generations? No one has been able to do so far. As far as I know, even the so-called epigenetic inheritance mechanisms that one reads about all the time fade off within a handful of generations. 10 maybe at the most. See, the problem here is genetic mutations occur over a time scale that you know. Selection of the kind that we know occurs within a time scale that we know. Anything that occurs over time scales much shorter than this is unlikely to have any effect other than fluctuating effects which you get all the time from the environment, temperature changes, rainfall changes, pH changes. You see the point. But if they're long lasting, and I think this is an empirical question. This is not a matter of philosophy or belief. Empirically, if you can show genetic information jumping not just across one generation, which we know, a few generations, which we know, but a great many generations, then that will have to be taken seriously. No. Please. No. I mean, I'm sorry, I'm throwing up my hands. That's precisely what does not happen. The idea is that the environment acts purely as a selective agent, not as an agent that stimulates mutation. So the environment does not have target genes in its mind and says, okay, you mutate in this way if you want to live with me. No. Exactly. So this is the principle of natural selection. Mutations occur at random. Some mutations are advantageous, some are harmful, some are neutral, as you saw. In general, the advantageous mutations spread. In fact, that's the definition of advantage. The harmful mutations are thrown out. Now, if you come back after a long time, you will find that the descendants of those that had the advantageous mutations are around now. But that's not because the advantageous mutations, for some reason, occurred with a higher probability than the other. Not at all. So the environment didn't cause the mutation. You get the point? Yeah. Yeah. No. That's the whole point. The whole point is that it's not targeted. Whole point is that it gives the appearance of being targeted. The first human being who made that spoon was doing that. He was mutating that piece of iron or whatever there was, or maybe even earth, which he was molding to a shape. He was targeting it to give rise to a particular. There's no target, no targeting, no design. Thank you. You're raising again a very fascinating question, but I really don't have the time to address that. I'll tell you this put this very briefly. Cancers are an example of how natural selection favors only the individual, the particular individual which is under selection at that moment. So in the case of organisms like ourselves, natural selection favors us. In the case of cancer, the individual suddenly becomes the individual cancer cell. You get the point? So it's a little bit like the cancer cell having no regard for the rest of the body, putting it figuratively, which it uses simply as a source of nutrition, as a source to be manipulated for its own use. Right? And therefore, it too evolves. And now, any number of examples of these drug treatment leads to the evolution of drug resistance in cancers and so on and so forth. In fact, it's not very different from what antibiotics do to bacteria. So you can think of the evolution of cancer cells from a natural selection point. Yeah. But they are like, you remember that, which example did you take? The virus example, I forget which one. Ultimately, they are doomed because they don't have a way, as far as we know, of going from one host to another. Once the individual human dies or the animal dies, the cancer cell is gone. By the way, there's an interesting exception there. I don't know how many of you know this, which is a viral cancer in the Tasmanian wild dog. It is this thing called the dingo. There it apparently moves from individual to individual by contact, which is a fantastic kind of test case for cancers to take on a life of their own. But I think human intervention there has seen that they don't spread much and so on. You should look it up. I'm easy, but I think the director is getting nervous. Come again. I didn't get the last part. What do you mean by good for scientists? Oh, it gives a job for scientists. Absolutely right. Absolutely right. Absolutely right. Yeah, I agree with you.
Thank you very much.
Very thoughtful. Thank you. Extremely thoughtful gift.
Thank you very much. That's our signature.
I hope you didn't mind my mild joke at your expense.
Bye.
Please do. It's a pleasure. They go out. So, oh, I was going to walk away with this. Thank you.
Outstanding.